Now, if you're a sci-fi fan like I am, you know, a Trekkie or a Star Wars nerd, or you just love anything sci-fi fantasy, you know, you'll have heard, you know, in a Star Trek or whatever movie, come across some new planet, and they'll talk about carbon-based life forms. Carbon-based means that predominantly the predominant atom and all the molecules that make up that living organism or any of the living organisms on that planet are based around carbon, hence carbon-based life form. Nowadays they refer to it as an organic biology, an organic organism. Now here, organic doesn't mean a couple of hippies out in their garden. No, organic means carbon-based. Think about it. Organic chemistry that you'll take your junior or senior year is going to be based on how molecules that are based around carbons interact. Carbon is one of the chief atoms that make you up. Make up your dog, your cat, the tree outside, the grass, every living organism on this planet. We find carbon to be the central atom in every protein, carbohydrate, lipid, the nucleic acids that make up your DNA and RNA. Everything is based around carbon. Why carbon? Well, it turns out carbon's unique. Its shape gives it some very peculiar functions that very few other atoms can match. They may be able to match one or two characteristics, but not all of them. Carbon, its valence shell only has 4 electrons. It means it has 4 openings if it wants to be stable. It means it can make up to 4 covalent bonds. Single, double, triple, doesn't matter. 4 bonds. Carbon also likes to interact with itself. So you can start linking carbons together, carbon, 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 carbon chains. That's what carbohydrates are. We call them sugar. It's what the lipids, the fats, are long chains of carbons. You've probably heard of the term hydrocarbons. Hydrocarbons are large macromolecules. Macro meaning really frickin' huge. These macromolecules are composed primarily of carbon as the central chain, and then hydrogens linked to every carbon. 2, or at least 1, depending upon the shape of the chain. You take these hydrocarbons for our bodies or whatever biological organism that made them, to link all those carbons together took energy. So between every carbon, 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 carbon bonds took energy to form. So it is now there's energy stored in that bond. Well, we take these hydrocarbons and we start breaking those bonds very quickly. We start to release the energy. Release the energy as heat, as we see here, flame. It's this quick release of the energy. Not a slow, methodical, but a fast release is what gives us the heat, the energy to power cars, to power energy or electrical substations, electrical generating plants. You take one of the hydrocarbons we see quite often here, methane. The central atom is carbon. Methane, CH4. Because of its shape, the carbons are going to form this weird tetrahedron. Tetrahedron means a 4-sided pyramid. And if you look at it and you squint, this hydrogen to that, and back up, 1. this one to this one, then back up 2. Here to here to here, 3, and then down here, 4. Carbon wants to form that tetrahedron shape. They will always form that shape. And remember, shape dictates function. So you start taking these tetrahedrons and you start linking them together like you did up in chemistry. CH4, methane. C2H6, ethane. How does this work? Well, look. 2 tetrahedrons inverted towards each other. C2H6. One of the electron-electron bonds is going to be here, the carbon-carbon. So you're going to be 2 hydrogens less, hence the H6. But what happens when you form the double bond, like you see here in ethene? Well, instead of being 2 inverted tetrahedrons, you now have a flat plane, kind of like a sheet of paper. Shape dictates function. Ethane, even though it's relatively close to ethene, C2H6, C2H4, ethane, ethene will react different in every chemical reaction. Shape dictates function. And what's interesting, you take these carbon chains and you start linking them together in weird ways. Cyclopentane. Penta means 5. 1, 2, 3, 4, 5 carbons. Notice it's C5H10, the maximum number of hydrogens present. Cyclo, because it forms a ring. Cyclohexane, 6 carbons. 1, 2, 3, 4, 5, 6. You start to throw in some alternating single and double bonds. 6 ring becomes benzene. Cyclohexane and benzene, only difference, half the number of hydrogens. Couple of double bonds. So what you see here, it's inverted to inverted tetrahedrons, and it just keeps repeating over and over. Whereas here, inverted tetrahedrons, flat plane, inverted tetrahedrons, flat plane, blah, 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 blah. Different shape, thus cyclohexane and benzene act completely differently. Hey, sometimes you can just pull out a hydrogen or pull out a carbon. Now you have something completely different. Now we have to be cautious here. Some of what you learned in chemistry comes into play here. Shapes of the molecules dictates functions. Look, butane, isobutane. 1, 2, 3, 4 carbons, 10 hydrogens. 1, 2, 3, 4 carbons, 10 hydrogens. These, while they have the same structural or the same formula, C4H10, they look completely different. They refer to these as isomers. Isomers have the same constituents but look completely different. Different shape, different functions. We find that isomers fall into 3 categories. Structural, as we see up here. Structure. Straight chain with a carbon off of it. Butane, isobutane. Completely different chemicals. Completely different compounds. Or it could be geometric. You have the flat plane of a carbon-carbon double bond. CH3, CH3, both on the same side, pointing in the same direction. Or one up, one down. One above the plane, one below. Cis, trans. Or sometimes you have what is referred to as the mirror image. These are called enantiomers. Enantiomers are going to cause problems. Sorry, I skipped ahead. Left hand, right hand. Your left hand, right hand look exactly the same except they're mirror images. Left hand, right hand don't act the same. Now let's look a little closer at this. The cis, the trans. Here, As before, we talked on a different slide in the last lecture. Red balls, oxygen, white balls, hydrogen, black balls, carbon. So you take this long chain of the black balls, the carbons, you add hydrogens across them, and then put oxygens. Boom, boom. But notice right here, this gap. This gap where you don't have all the hydrogens there means there's a double bond there. Sometimes that double bond will allow for a straight chain, but more often than not, it means that long chain of carbons is going to get a kink, a bend. Trans, everything's on the same. Are on opposite sides. Cis, everything's the same. Now, when we get into talking about the membrane chapter, and we're talking about phospholipids and the lipids and the fats, what we're going to see is that these lipids are composed of long chains of carbons, like aldehydic acid and oleic acid. If there are, every carbon has its maximum number of hydrogens, that's referred to as saturated. But sometimes those hydrogens go away and you get a double bond. Means you do not have the maximum number of hydrogens and you have the potential for a bend. This is unsaturated. You've probably heard this in some nutrition course, saturated, unsaturated fatty acids. That's what they're talking about here, oleic and aldehydic. Does it have all the hydrogen possible? Are some missing unless you have a double bond? Shape dictates function. The two acids will act differently. The enantiomers that I skipped ahead on, sorry about that. Mirror images. D-alanine, L-alanine. They kind of look the same, just, when you're looking at yourself in the mirror, you see everything in reverse. Same here. The metabolic or chemical reactions that occur either in a test tube or in a living cell generates alanine. Alanine is one of the amino acids you need for your proteins to make polypeptides. L-alanine is the correct shape to actually make a functioning protein, a functioning enzyme, whereas D-alanine, well, it still has the same constituents, but it's the mirror image. This doesn't do anything. This cannot be used in a living cell. Now, this slide is very important. I'm going to warn you now, you need to know this slide. This slide will be on the test. This slide will be in almost every lecture from here on out. A long strand of carbons, like we saw with oleic acid, actually does nothing. It sits there. It took a lot of energy to link all those carbons and hydrogens together, and it does nothing. It doesn't interact. It's nonpolar, so it can't interact or do anything unless you add some attributes to it. Attributes in the form of these functional groups. Functional groups, by their name, gives a carbon chain, whether it's a cyclic chain or a straight chain, gives it an ability to interact with other molecules in close proximity. You look at these molecules, the hydroxyl, an OH, an O and an H, negative-ish, positive-ish, means it's going to make whatever is on it polar. Means it can interact with water, it can interact with other ions, other charged molecules, charged atoms. Or a methyl group, CH3. It's nonpolar, but this can interact with other things out there, but not in such a polar hydrogen bonding method that the hydroxyl will. Carboxyl. Carbon, oxygen, COOH. COOH. The hydroxyl, the OH, and that double bond oxygen will have charges. They will ionize. They will give it a polarity. What's also interesting is that the hydrogen right here in this hydroxyl is easily released into water. It will increase the concentration of hydrogens in the solution. Think back to chemistry. High levels of free hydrogen in a aqueous solution means that that solution is now acidic. So, high levels of carboxyl means it's going to make an acidic environment to whatever molecule this functional group is attached to. The amino, they call it an amino because it has a nitrogen right there. The amino, NH2, easily accepts the hydrogen released by a carboxyl. It pulls it in. Why do they call it amino? Because it's the base of an amino acid. Instead of a carbon, it has a nitrogen off to the carbon. So instead of carbon chain, it's nitrogen there. Phosphate, PO4. 1, 2, 3, 4. 4 oxygens. These hydrogens will be released, and you now have a polar molecule, or at least the environment, the area that this phosphate functional group is in, will be acidified. And then finally, sulfhydryl. That sulfur is going to come into play when we talk proteins. A disulfide bond means that it's going to hold proteins together. Now, you've heard me talking about hydrogen bonding in these molecules, the release of a hydrogen here, release of a hydrogen there, and you can see it right here. These hydrogen bonds allows thymine to interact with adenine. Oxygen that is negative-ish, a hydrogen that is positive-ish. Hydrogen that's positive-ish, nitrogen that is negative-ish. Remember, no energy to make this interaction occur, thus it takes no energy to pull them apart. Look at what we're looking at. Thymine, adenine, guanine, cytosine. What we're looking at are DNA bases. DNA is highly stable. We're isolating parts of DNA from mammoths that have been frozen for, 50,000 years, mummies that have been desiccated for almost 8,000 years. We're getting, some DNA out of them. Why is this so stable if it's only based on hydrogen bonds? Yes, phosphodiester bonds holding the different nucleotides together in a long chain, and then the two strands, the two chains held together form the double helix hydrogen bond. Where one hydrogen bond is weak, billions and billions of them holding the two strands together. Well, that starts to add up.